皆さん、こんにちは。Hello to everyone in San Diego。LINK-J のライブウェビナーにご参加をいただきありがとうございます。LINK-J の高橋です。本日は LINK-J と UC サンディエゴジョイントウェビナーシリーズ第12回 with 慶応大学再生医療と計算神経科学における最新の展望セッション5、脳オルガノイドを用いた最新の研究をお送りいたします。本日は画面で示しておりますプログラムに沿って進めてまいります。この UCSD とのウェビナーシリーズも12回目となりました。世界のトップクラスの研究者の方々に最先の研究をご紹介いただく非常に良い機会となっています。これも UCSD の和賀さんをはじめ、多くの研究者、関係者の皆様のおかげと思います。どうもありがとうございます。さて、今日は脳オルガノイドをテーマとしています。オルガノイド研究はここ数年、創薬への応用であったり、分子メカニズムの解明など、さまざまな活用が報告をされています。そのようなホットなトピックを、今日は岡野先生とコーエンバーク先生がモデレーターという豪華なセッティングで、お送りすることになっています。私も大変議論を楽しみにしているところです。どうぞよろしくお願いいたします。なお、このご質問等あります場合は、ウェビナーの Q&A 機能からお願いをいたします。時間の関係上、すべての質問にお答えできかねる場合がございます。また、本イベントでは、ウェビナーの同時通訳機能、英語字幕をご利用いただけます。字幕は市販されているアプリを使用しており、LINK-J、UCSD、慶応大学は語訳、欠落等、字幕に関する一切の責任を負いかれますので、どうぞご了承ください。なお、ウェビナーの録音、録画、画面のキャプチャーは固くお断りをしております。最後になりますけれども、ウェビナー終了後に表示されるリンクより、アンケートフォームにお進みいただき、皆様のご意見、感想をぜひお聞かせください。皆様の役立つセミナーを開催したくご協力をどうぞよろしくお願いいたします。それでは、UC サンディエゴ国際アウトリーチシニアダイレクターの和賀さんより、イントロダクションとモデレーターの紹介をお願いいたします。では、和賀さん、よろしくお願いします。
はい。日本の皆様、おはようございます。サンディエゴから視聴の皆様、こんにちは。UC サンディエゴで国際アウトリーチのシニアディレクターを務めております、和賀でございます。コロナ禍で渡航制限がかかる中、始まったジョイントウェビナーが今回で12回を数えるほどになりました。このうち慶應義塾大学とのセッションは今回で5回目となります。いつもご指導していただいております岡野先生並びに、毎回きめ細かくサポートしてくださっているLINK-Jの方々に心よりお礼申し上げます。今回より慶應義塾大学とのセッションでは、コーエンバーグ先生が UC サンディエゴ側のモデレーターを務めてくださることになりました。コーエンバーグ先生はバイオエンジニアリングの教授であり、Institute for Neural Computation のコーディレクターも務めておられます。先生は UC サンディエゴのみならず、近隣研究機関の脳科学者とも緊密に協力されており、モデレーター役をお引き受けくださったことを大変嬉しく思っております。今回 UC サンディエゴより参加いただくアリソン・モートリー先生ですが、実は昨年5月に日本橋でハイブリッドのシンポジウムを開催した際、アンケートへの回答の中に、ぜひモートリー先生のご講演をお願いしますというお声がありました。先生をお招きする機会を探っておりましたが、岡野先生もモートリー先生をよくご存知ですし、島田先生というぴったりのカウンターパートにも恵まれて、今回のジョイントウェビナーを開催する運びとなりました。まずは慶応の岡野先生よりご挨拶をいただいた後、コーエンバーグ先生よりご挨拶と、最初の講演者のご紹介をいただきたいと思います。岡野先生、どうぞよろしくお願いいたします。
Hello, everybody. This is Hideki Okano from Keio University, and also I'm the chair of the board of the LINK-J. And as Takashi-san has mentioned that, so LINK-J and UC San Diego has performing very collaborative work, and so this webinar is 12th webinar between the LINK-J and UC San Diego. Actually, and Keio University and so UC San Diego has signed up MOU in 2017 and before the COVID begins. And that is, we started from the UC San Diego and the health school and K University School of Medicine. Later, both of the engineering and so in KO Faculty of Science and Technology joined. Now we have very integrative science aiming for the human health. Okay, so now what's the theme of this webinar? We have discussed about it. So actually, so in KO and UC San Diego, in the both schools are very strong in the IPS-based regenerative medicine and also neuroscience. And what's important topic that satisfies both sides of it, that is the brain organoid. We thought about it. So actually, so I have been collaborating. So Dr. Alison Motori for many years and had published a couple of papers. And in my lab, and so Hiroko Ishii, today's speaker, has published a very nice paper about recapitulating the Alzheimer's disease using brain organoid. Now this is very important to make share information and talk about the future collaboration each other. That's why we started the topic of the brain organoid today. Anyway, please enjoy this webinar today. Thank you very much for your attention.
Hello, everyone, and it's so exciting to have this webinar series. So LINK-J is a wonderful opportunity to connect between our colleagues at Keio University and others in Japan, and then here in San Diego, UC San Diego. It so happens that we have really great synergies in life sciences between two universities that have been really come forward in this series of initiatives. And so today, again, we have a really exciting webinar on new horizons in regenerative medicine and computational neurobiology. And both universities have a very strong programs, both in regenerative medicine, stem cell research, organoids, and with clinical applications. What is exciting is the work by Alison Motori really bridges this regenerative medicine and approaches of basic stem cell research and organoids to the cognitive level, and it's really able to so in terms of understanding development, both children and basically or entire lifetime. So Dr. Motori, he is a professor of pediatrics and cellular molecular medicine, also co-director of the stem cell program here in San Diego. And today he'll tell us some exciting research on modeling human neurodevelopment with brain organoids. And so Alison, please.
Thank you so much for the invitation, and thank you, Gord, for the introduction. I'm happy to be here and share some of the work that we are doing. My lab has a large repertoire of applications for brain organoids. So I decided to focus on a story about a disease called Pitt-Hopkins syndrome, and I will use PTHS as an abbreviation for that. And that story started a couple of years ago when we were approached by a family foundation to see if we can use the brain organoids to learn a little bit more about Pitt-Hopkins and to develop a better treatment. So I think this is it's a good illustration of how this technology can actually move forward if you have the right tools in hands, and you'll see in a minute why. So first of all, I would like to say that this work was mostly done by Dr. Fabio Papis, who is a colleague and a friend from Brazil, who decided to do like a sabbatical in my lab. And Fabio has someone with Pitt-Hopkins in his family. So he decided that this would be something that will bring motivation for him while here in US. So this is a project that we designed and decided that he would execute with other members in the lab. So what is Pitt-Hopkins syndrome? Most often, they are under the umbrella of autism. So some of the kids that has a mutation in a gene called TCF4 can be diagnosed with autism. But there are quite severe cases that highlights the spectrum of this disease, motor delay and generalized hypotonia. We see quite often. So these individuals might have like some initial normal life, and then, I mean, they start to regress and they end up in a wheelchair. Most of them will not walk or talk, actually. So they have this hyperventilation or apnea, quite quick breathing that might lead to seizures. We don't know if this is correlated or if there is a causation here between those two, so it's still unknown. As I mentioned, most of them has absence of speech, so they cannot talk, they cannot express themselves. There are lots of stereotype movements, rocking or jumping, or I mean, in the ones that can do that. Self-inflicted aggression is something that we see as well, and intellectual disability. And another thing that is also quite important for the families is this severe constipation. From time to time, the families needs to take these individuals to the hospital so they can deal with this constipation. So this all suggests that the gene is quite important for several aspects of neurodevelopment, and is not restricted to the brain, but is actually expressed in all cells of the body. So this is a transcription factor that we don't know much about it. We are still revealing what are the downstream targets of this factor. We know some, but it still remains to be discovered what are the target promoters or target genes for TCF4. All right. So we and others have decided to try to model that. And of course, I mean, there are a couple of mouse models that were made by mutating the TCF4 gene in mouse, and there was not much phenotype. So this doesn't surprise me. Several of those genes that are important in early neurodevelopment do not show phenotypes if you knock them down or if you mutate them in a mouse model. So that was not a big surprise. So the mouse models, they can walk, they can have like a normal life, normal lifespan. Maybe there is slightly behavior alterations depending on the methods that you use that you can actually detect those. But overall, I mean, the brain is normal, and the behavior of the animal is quite normal, which does not make it a good translational model. And that's why we decided to focus on the human model. And to me, that's one of the perfect application for a brain organoid. And as you know, I always would like to highlight and acknowledge the contribution of Yoshiki Sasai to this entire field. I consider him the pioneer of brain organoids, and specifically this paper here in 2008, that's exactly when I was establishing my lab. And I remember reading it and seeing all those images showing this very nice anatomy that he was able to generate from human pluripotent stem cells in suspension. And I said, that's exactly what I want to do in my lab. This is what I think is a perfect disease modeling for this type of conditions. So we decided to take advantage of iPSCs. So we reprogram cells from patients with Pitt-Hopkins carrying the mutation and other mutations. And then we decided to make human brain organoids from that. And since the original publication from Dr. Sasai, there has been lots of variability or variations in these protocols. And I like to use this slide just to highlight some advantages and disadvantages of the protocols that you're going to use. And very didactically, we divided in unguided differentiation or guided differentiation, just to make it easy. The unguided one are the type of differentiation that you have multiple brain regions in the same unit. Because of that, you have a nice representation and diversity of cell types, but you lose their reproducibility. This type of protocol is more heterogeneous, give rise to different units and becomes harder to do disease modeling unless you have a very dramatic phenotype. I think most of this field is now inclined to use variations of the SASAI protocol, where we use factors to guide the differentiation towards the specific brain regions, and for example, highlighting here cortical brain organoids. And there's a tendency also to study circuitry by mixing or fusing different types of brain region-specific organoids to create what we call now assembloid, for lack of a better nomenclature. For this specific project, I'll be using guided differentiation towards the cortical fate, mostly because we knew that the cortex is one of the regions that is implicated in autism and affects several other neurodevelopmental disorders. I also like to highlight the limitations of this technology. You hear in the news about mini brains being generated in the lab, and gives you the wrong impression that you have an entire brain, a petri dish, which is not true. I think most of you know that what we have is just a tiny tissue with a couple million cells in there. And these are quite immature cell types. The organoids are not vascularized. We don't have all the diversity that we want in terms of cell populations. And it's still, we are optimizing the protocol, so we don't have all the ideal conditions for this to become like something quite reproducible. And the translatability of the organoids are still building momentum. So we have like a couple of examples, and I'll give you one now of data that can be translated or easily translated into humans. All right. So this is our modification of the SASAI protocol, and we have this recipe now in the lab, and we start with dissociated human pluripotent stem cells, could be either iPSCs or human embryonic stem cells. We like to grow everything in ideal conditions. These are all media that are chemically defined, so we don't have variability in there. And after that, we use the dual SMAD inhibitors to induce the neuralization and the cortical fate. And then we stimulate with FGF followed by EGF, so we can get a nice proliferation of the cells. And after that, we just remove the growth factors and let the cells differentiate and mature. So we start from single cells, and we end up with about 5 million cells in some of these structures. And some people ask me what kind of cells you have. And so it really depends on what is the stage of the organoid. So this is quite dynamic structure. We start with pluripotent cells, and we end up with 5 million cells in there. And this is a snapshot at 4 months. At 4 months, we have our pool of progenitor cells that continues to produce glutamatergic excitatory neurons. At 4 months, we start to see that some of these progenitor cells become gliogenic, and they will mostly produce astrocytes. And that's exactly the time when we see the peak of synaptogenesis in our organoids. We also see a discrete population of GABAergic neurons, and we like to say that these are not yet inhibitory neurons. They express the markers for GABA, but we know that they only become inhibitory a little bit down in the development when these organoids are about 6 to 8 months. So these cells start expressing these markers, but not quite yet function as GABAergic neurons. Nonetheless, I mean, I think this is a nice illustration to see that we have all the major components of the cortex being represented in there. And as you age the organoids, the diversity of the cortical layering start to appear as well. So we start to see more and more markers of different cortical layers. Although over time, you kind of lose some of the nice anatomy that we have in the beginning. And we can discuss later why this is the case. But nonetheless, so these are the cortical organoid. We have a representation here from the parent. We always work with a related control, someone that has the same sex as the proband, such as the biological dad or mom, or a non-affected sibling. In this case is the parent, and you can see you can make nice cortical brain organoids from them. And what becomes immediately clear was that all the time when we apply the protocol to iPS-derived organoids coming from the Pitt Hopkins patients, we see like that they grow slow, and the organoids show several anatomic malformations, and I'll touch that later on. And they end up smaller compared to the control. So the size of the organoid is actually a fraction of their parents. So we also generate other brain regions. So this is our inhibitory pool of neurons, and we have a protocol for that as well. And in this case, I mean, we have larger organoids coming from the parents, and smaller organoids coming from the probands. And this is true across all the patients in all the cell lines that we try, which is about, I think we derived 10 iPSCs from the patients and 10 for the controls. And most experiments, we use at least 3 or 4 patient lines and control lines. So here you can see on this Petri dish that you can actually see a naked eye the size of these organoids and how tiny they are compared to the organoids derived from the patient cells. So when we slice these organoids to do some immunostaining, that's when we start learning about what are the major differences that we have. And so here is a normal organoid, the parental organoid. So you have like several of these rosettes. And in this rosette, you have population of progenitor cells marked here by SOX2, and some of the neurons marked here by magenta. So these rosettes are abundant in control organoids, but we barely see them in the patient organoids. So they almost don't form the rosettes or very tiny rosettes, and this SOX2 population will not actually grow. So this is the quantification at 4 weeks. You have plenty of progenitor cells, and as you would expect in the controls, the population of SOX2 reduces. But compared to the Pitt Hopkins, that doesn't have that many SOX2 positive cells to begin with, and they continue to reduce over time. So the absence of SOX2 or proliferation in the progenitor cells was something quite dramatic to see in these organoids. So this is the quantification of number of the rosettes that we see inside of the organoids. Again, in the parents, as they move from 2 to 4 weeks, the number of rosettes grows, and then kind of a plateau over time. And in the proband, we barely see rosettes, and they just die over time. So it's a nice illustration of how something very simple can be quantified from these organoids. And each marker here is one of the patients. You can see a ball, a square, a triangle. So these are all different iPS cells, different individuals. And we usually work with at least 2 clones for each one. So you can see how robust is the data. And we feel that it's important to do that multiple times because sometimes 1 or 2 patients doesn't seem enough for us to cross the variability of the experimental protocol as well. So having multiple patients, I think it's crucial. All right. So why these SOX2 positive cells die? So this is another thing that we notice. The SOX2 positive cells, they have early markers of senescence. So we see that very clearly by staining with the P16. So the number of, for example, dying cells is much higher in the proband compared to parental cells. So at this stage, we were wondering if those observations were actually true or some kind of an artifact. And then that was a key point in our process because one of the patients died, and the family was kind enough to donate the brains of the patient to the science. So now we have the same iPSCs that were derived from the patient, and we have the patient postmortem sample. So we could do some cross-validation. And this is, for example, when we quantify the number of senescent cells in the postmortem tissue. If you use a age-matched control, you barely see cells senescing there because these are young people, but it's quite large in the postmortem tissue that we're able to see. So that was a nice validation. So we also noticed that if you do have a defect in SOX2 positive cells, the proliferation of these cells should affect the cortical layering or the diversity that we see in the cortex. So we quantify specific cortical markers. And here I'm showing like a couple of them. But I think it's easy to visualize, for example, CTP2 in magenta. We have lots of dots in CTP2 showing like CTP2 positive cells. These are, again, specific cortical layers 5 and 6. In week 6 and weeks 9 and 10, you just increase that population because those progenitor cells will differentiate into that. And you barely see that in the Pitt-Hopkins organoids. So again, you quantify that, you see that increases the number of CTP2 positive cells with time, but the proband doesn't show much, and it might have like a slightly increase over time, but there is no even comparison. I mean, you can see it, you don't need the statistic to show the difference here. You can see it by naked eye. So if that's true, we should see a lack of CTP2 positive cells in the cortex of the Pitt-Hopkins individual. So we turn back again into the postmortem tissue and we compare. We look for CTP2 positive cells. That's a specific cortical layer. And we see that in the control. These are the red dots here, but we barely see it in the postmortem tissue. We see like some clusters of cells, but that's it. I mean, we don't see a nice cortical layer of CTP2 positive cells. Again, suggesting that these brains are missing such an important cell type that was not there to begin with. We also use the postmortem brain tissues to quantify some of the genes that might be implicated on that, and was reassuring to see that TCF4 was downregulated by 50%, consistent with haploinsufficiency that we would expect for this condition. Other genes such as CNT and AP2, which are target of TCF4, is also downregulated in others. And it's interesting to see SOX2 upregulated, even though the number of SOX2 positive cells it's lower. So our interpretation here is that this is a tendency of the system to kind of compensate for the lack of cells that we have during development. All right, so if this is a haploinsufficiency, we still have a normal and healthy copy of the gene inside each one of these cells. And if we can boost it up, the activity of that healthy allele inside the cells, we should be able to perhaps level up the TCF4 levels inside the cells and maybe rescue some of these phenotypes. So we decided to take this approach using a CRISPR activator strategy. So these are a strategy where we use a guided RNA against the TCF4 promoter, and we assemble the complex using a dead caspase 9. So there is no cleavage in the DNA, so there is no nick in the DNA. The only thing that the CRISPR would do is to form this complex to activate the gene. And of course, I mean, with this strategy, there is no way to distinguish between the mutated and the health allele. So we're going to have an overexpression of both copies of the gene. But our hope was that maybe having like enough of the Y-type gene could revert some of the cellular phenotypes that we saw. And so here is just some examples of the guided RNA. The TCF4 gene is tricky because it has several isoforms, and nobody is 100% sure which kind of isoform is important in which cell type. But using the postmortem tissue, we focus on the TCF4 isoform B, which seems to be highly expressed in the brains and also in our organoids. So we went ahead and designed a couple of guided RNA to target that promoter. So we could stimulate the expression, again, using the CRISPR activator. And this is our protocol. We use the CRISPR in a lentivirus to infect the cells very early on during the early formation of these clusters of cells just before the neuroinduction. And we kept the lentivirus inside the cells throughout the protocol. And so here is the parental morphology of the organoids with a scramble guided RNA, so it's not activating anything. Here is the probands with the scramble guided RNA, so we see all the malformations and the small size of the organoids. And here is the same cells, but now with the guided RNA targeting against TCF4. So you can see a nice rescue of the size and morphology of the organoids. So we also look for immunohistochemistry to see if we can see some kind of a rescue of some of these cell populations that I mentioned earlier. And so here is the number of SOX2 positive in green, and the number of neurons in magenta. And some of these malformations that we see in the organoids are actually caused by clusters of neurons that cannot really migrate and form anything. So they become a scar inside of the organoid. So this is one example. This is another example. The organoids kind of show this aberrant morphology caused by these clusters of neurons. But once you use the CRISPR activator, you fully rescue all the morphology and all the cell types, including these structural alterations that we thought that will not see a rescue for those. We also see rescue at the molecular level. So show you some examples. Here is the overexpression of TCF4 on the blue light bar. Again, because we are overexpressing both the mutant and the correct allele, we see more than the controls. GAD-D45G is a downstream target of TCF4. It's downregulated in the patient here in dark blue. Upon the CRISPR activator, we see that the expression is rescued. TCF4 do also inhibit CDK and 2A. So we see overexpression in the proband compared to the controls. Upon CRISPR activator, again, we are able to repress that gene. So at the molecular level, it seems that we are fixing things. And at the cellular level, we are also fixing some of the most dramatic phenotype. So this is all good, but the CRISPR activator strategy is still tricky to move into clinics because it requires like 2 viruses to infect the same cell. And there are also concerns about potential off-target effects on this strategy. So we decided to do a different approach. So here we would try to rescue the expression of TCF4 by a simple AAV9 delivery carrying the cDNA of the gene. The challenge here was to choose the right promoter because most promoters would upregulate TCF4 to a level that was not physiological. So how we decided to come around was to use micro E5 boxes or cassettes clone upstream to a minimal promoter upstream to the TCF4 isoform B cDNA. So these micro E5 motifs are the sequences of DNA that TCF4 binds to activate or repress downstream genes. So our idea was, so if we clone them upstream to a minimal promoter, the TCF4 that would be inside the cells can self-regulate and control the normal levels of TCF4 inside the cells. So we didn't know how many of these micro E5 boxes were necessarily. So we try with 5, 4, 3, 2, 1, and you can see that by reducing the number of TCF4, you increase the expression. So we figured out that probably like 5 is something close to a control, and we should use that. So the TCF4 will self-regulate, create like a feedback loop to self-regulate inside the cells. And that allows us to infect more cells as well. We don't have to be limited to neurons, so we can infect glia cells and any other cells as well. So we quantify again the level of TCF4 and the level of some of the downstream genes using 5, 12, or 6 micro E5, and they all seem to work very nicely. So then the experiment was to use the AAV9, but now instead of early on in the protocol, we could add them a little bit later, once the phenotypes is already there, to see if we can have enough to rescue some of these phenotypes. So here's the morphology of the parental organoid, big, full of rosettes, as I mentioned. And here is the proband, small. We don't see many rosettes in there, in half of the level of TCF4 here in red. But after the AAV delivery, you rescue the expression of TCF4. You now can see TCF4 in every single cell. You rescue the number of rosettes, and you rescue the number of SOX2 positive cells. So here's some immunostains to prove that. Again, control here on the left, proband in the middle with a control vector. And after the transduction with this AAV, the rescue of the SOX2 positive cells and the morphology and size. We also look for the CTP2 positive cells that is lacking inside of the proband organoid. But again, I mean, we see a rescue back after the AAV delivery. So this is all quite fantastic and allow us to be confident on plating these organoids on top of a multi-electrode array, so you can record the activity of these organoids. And this is just a summary of some of the data that we can get from control organoids here in orange. You see that over time, the activity goes up and then kind of a plateau here. Same thing with the number of bursts that we can get from the multi-electrode array. The patient cell lines, they have much lower than that, so they never really cross any significant level here. They kind of a plateau. But after the AAV delivery, then yes, we can see a rescue. It takes a little bit of time, most likely because the cells are still being produced inside of these organoids, especially the CTP2 positive cells. But after a while, we see a full rescue and they start to behave similar as the control cells. So we see this nice peak again of electrical activity. So our proposed model for this condition now, and again, only based on the human model and with some validation of the postmortem tissue, it is that we have a deficiency on the pool of neuroprogenitor cells. And I haven't shown, but this work was recently published, and you can read. We figured out the molecular basis for the lack of progenitor cell replication here, and this is due to a low level of SOX3 positive cells that can be activated by the Wnt signaling here. And this causes a lack of intermediated progenitor that produces a lower number of cortical neurons in the patients compared to controls. So this is our working model. So moving forward, I mean, I think we are quite confident that we have a decreased proliferation that changes the composition of the cortical neurons, causing disruption at this network level. And we can rescue the TCF expression, both using an epigenetic method, such as CRISPR activator, or an AAV vector that provides a gateway for future therapeutic strategies for Pitt-Hopkins and other TCF4-related conditions. TCF4 is also implicated in schizophrenia. Is one of the top hits for GWAS is on the TCF4. So we might have a way to, by working with a rare syndrome, we might have a way to explore that for our larger problems such as schizophrenia. So I'm happy to say that this technology was recently licensed by Mazi, which is a spin-off from Ultragenyx. It's a company that deals with gene therapy here in US. And we just received our data back from the monkeys showing that the toxicology is related to the AAV, so there is no problem with the TCF4 expression. And we are finally, or we're hoping to move with that with an FDA IND and start recruiting perhaps by the end of the year to do a proper clinical trial. So I'll finish here by thanking the members of my lab. As I mentioned previously, this was mostly led by Fabio Paps, who was a visitor in the lab, with the help of many postdocs and grad students in the lab. And full disclosure, I am a co-founder of Tismo. It's a company that works with genetic models of autism and brain organoids to find better therapeutics for this population. And my funding agencies are mostly NIMH and CERN here from California. And I'm happy to take any comments and questions. Thank you.
Thank you so much, Alison. Wonderful, really great research. So there are a few questions, and there's one on the Q&A. We can have a look at this. I can read it for you. So the question so the GAD45G has been shown to be a gene required for axonal elongation in a study led by Professor Okano. So it was interesting to see that it is also involved in this disease model of organoids as well. So the question is, what did you focus? So why do you focus on GAD45G and measure its expression level?
Yeah, so the answer is quite simple. It's one of the few genes that is regulated by TCF4. So we use as a marker to show that we have enough TCF4 inside the cells to actually regulate or activate that gene. So we are still looking for other genes that are downstream to TCF4, but that is one that has been characterized by different groups, and everybody would agree that it is a good downstream gene. That was the reason.
I have an additional comment. As you know, this is GAD45G has human-specific deletion in the enhancer promoter domain. Maybe there's some human-specific regulation of this gene. So it's very interesting to see what's it doing in the human disease. And also, GAD45G by itself has ability to the neural axon extension. So in that sense, it could be related to neurodevelopmental disorders and psychiatric disorders as a readout of the TCF4. Could be the direct target TCF4. Maybe interesting to see what happens.
Yeah, no, that was a nice observation. I totally agree. Yeah, not only for Pitt Hopkins, but schizophrenia and maybe other conditions. Yeah, that's a good point.
Mm-hmm.
So very exciting. So one more quick question here. So it's so exciting that you can use those brain organoids not only for studying a very important disease, but you also have a means for validating some strategy for therapy. And you show this great work with CRISPR activators or AV promoters. Now, so since brain organoids are not quite the same as an in vivo because the system. So do you expect the clinical study to give consistent results with the very promising results that you observed with the brain organoids?
Yeah, you know, this is remains to be seen, right? Because it is an interesting model, but remember the model is recapitulating early stages of fetal, perhaps early postnatal. And so far we can rescue that, but the real gene therapy would happen in patients that are a little bit older. Maybe we can target the kids that have the diagnostic at 2 or 3 years, but most likely would be a little bit older kids, and their brains are way more complex than an organoid. So would be the AAV enough? We don't know. So recently we have been developing assays to try different amounts of AAV to know exactly how many cells we need to target to have a reliable response from a rescue. And depending on the condition, we are getting different results. For example, I also work with Rett syndrome, and if we use the same approach, 5 to 10% of correction seems to be enough. So you have like a decent rescue. So you actually don't need to target the entire network, which is interesting from the fundamental perspective. Why this is it? I mean, maybe there is a hub of circuitry that we are targeting, and that's enough. But we don't know yet for Pitt Hopkins. And I think some of these questions, if it will work, how much is the rescue? What are we going to rescue, right? How long does it last? Unfortunately, I think just by doing it in people that we're going to learn it.
Very exciting. And so it would be wonderful to see this panel. So that's great. Thanks again, Dr. Mitri. And so now we pass it on to our friends at Keio University.
Thank you.
Okay, thank you. Thank you very much, Alison. And so, Jared, okay, for a wonderful presentation. Okay, let us start from the Keio side. Let me introduce Keio speakers and Dr. Hiroko Ishii. She is an assistant professor in the Department of the Physiology, Keio University School of Medicine. Actually, she did her PhD work in my lab, and she did postdoc training in Washington, D.C., National Eye Institute, and so she did organoids modeling of the retinal diseases. And then she came back to Japan, about 5 years, 2016 or 2017, and so she joined the project of the modeling the Alzheimer's disease with brain organoid technique. And she last year she published a wonderful paper about Alzheimer's disease modeling with brain organoid. And she is now promoting that project, and very exciting results coming out. Okay, so Hiroko, could you start your presentation?
Can you see the slide?
Yes, no problem. Go ahead. Yes, please go ahead.
Thank you. So hello, everyone. I'm Hiroko Shimada from Keio University. Thank you for the introduction, Professor Kano. Thank you very much for allowing me to give a presentation today. I'd also like to thank LINK-J for hosting this webinar. I'm delighted to be able to tell you about our recent study. Today, I will be presenting on modeling Alzheimer's disease and tauopathy in brain organoid. Let me start with a brief background. Alzheimer's disease, or AD, is a progressive neurodegenerative disorder. And AD patients show brain atrophy and neuronal loss. The two major pathologies of AD are formation of amyloid plaques due to the extracellular accumulation of A-beta and neurofibrillary tangles due to the intracellular accumulation of tau. Patients with early onset familial Alzheimer's disease, or FAD, have mutation in amyloid precursor protein, or APP, presenilin 1 and 2. This fragment of APP shown here is in red is called A-beta and consists of 40 or 42 amino acids. A-beta fragment is excised by beta secretase and gamma secretase, and presenilin, one of the causative genes, is a component of gamma secretase. This slide shows various neurodegenerative disease models. Although many rodent models have been developed, they do not exactly recapitulate the human pathogenesis of the disease. In addition, access to patient brain tissue is limited. On the other hand, iPS cell-derived neurons have the potential to reproduce currently ongoing neurodegeneration, making iPS cells a promising approach for generating in vitro models of dementia. In particular, brain organoids have the advantages of high neuronal maturity and complex cell-cell interactions. Several groups have described AD phenotypes in neurons derived from iPS cells established from patients with AD. These models showed early AD phenotypes such as altered A-beta secretion, enlarged endosome, and increased phosphorylated tau. However, late AD phenotypes such as amyloid plaques and neurofibrillary tangles were difficult to exhibit in 2D culture. Here are the advantages of 3D brain organoid. First, brain organoids are structurally and functionally similar to the in vivo human brain. Second, complex cell-cell interactions can be observed in brain organoid. Third, neurons in the brain organoid are more mature than those in 2D cultures. Finally, brain organoids have a 3D microenvironment. Therefore, it is expected to reproduce A-beta and tau pathology more prominently in brain organoid. In this study, we generated brain organoid from AD patient-derived iPS cells and tried to develop AD model brain organoid that reproduce A-beta deposition and tau aggregation. Here are the main topics of today's presentation. First, I will present an improved method for efficiently generating forebrain organoid, which I will refer to as FBOs, from feeder-free iPS cells by controlling FGF2 concentration. Next, I will introduce the FAD model FBO using iPS cells derived from familiar Alzheimer's disease patients, and the tauopathy model FBO, in which tau P301L is overexpressed in FBOs by AAV injection. Finally, I would like to introduce our current progress on the single-cell RNA sequencing of the tauopathy model FBOs. Let me begin with the first topic. Feeder cells were conventionally used to maintain iPS cells, but recently, feeder-free iPS cells, which do not use feeder cells, have become mainstream. Although FGF2 is essential for iPS cell culture, and the medium for feeder-free iPS cells contains a high concentration of FGF2 of 100 nanograms per milliliter, I was concerned that the high proliferative capacity and undifferentiated maintenance ability of feeder-free iPS cells might affect the efficiency in differentiating into brain organoid. Therefore, I decided to lower the concentration of FGF2. When the concentration of FGF2 was decreased to 10 nanograms per milliliter, the morphology changes slightly as shown here. With high FGF2, the individual cells appear compact, whereas with low FGF2, the cells are flattened and gaps are visible between cells in some areas. The concentration of FGF2 was reduced stepwise from 100 to 30 and finally to 10 nanograms per milliliter immediately before differentiation. iPS cells cultured with 10 nanogram per milliliter FGF2 formed embryoid bodies, or EBs, that were more stable in size and morphology than those generated from iPS cells cultured with 100 nanogram per milliliter FGF2. These low FGF2 EBs formed multiple thick neuroepithelium-like structures. Although most high FGF2 EBs became cystic and had Q neuroepithelium-like structures. Measurement of diameter of EBs showed a large intra- and interbatch variation in the high FGF2 EBs, but less variation in the low FGF2 EBs. Next, we examined gene expression of low FGF and high FGF iPS cells and EBs. What we found is that forebrain markers such as FOXG1, SIX3, LHX2, and RAX were more highly expressed in low FGF2 EBs than in high FGF2 EBs, while several midbrain and mid to hindbrain markers were more highly expressed in high FGF2 EBs. The PCA shows that PC1 component represented the variation in gene expression between iPS cells and EBs, while the PC3 component was considered to represent the difference between low FGF2 EBs and high FGF2 EBs. Notably, low FGF2 iPS cells and high FGF2 iPS cells did not separate along PC2 component as well as PC3 component, suggesting that the concentration of FGF2 largely affects gene expression in EBs after initiation of neural induction. Moreover, immunohistochemical analysis showed that SOX2 positive neural progenitor cells positive for the forebrain marker FOXG1 were observed in low FGF2 neuroepithelia, while FOXG1 positive cells were not found in high FGF2 neuroepithelia. These data suggest that low FGF2 EBs acquired forebrain identity. These neuroepithelia were transferred to bioreactors and cultured in rotation. As a result, brain organoids containing deep layer neurons, superficial layer neuron, and astrocytes were generated, and a large number of synaptophysin positive puncta, a synaptic marker, were detected on day 84. In addition, MVP positive oligodendrocyte could be also observed on day 126. Thus, by controlling the concentration of FGF2 in the iPS cell culture medium, we were able to establish a method to efficiently generate forebrain organoid from feeder-free iPS cells. Next, I will introduce the F80 model FBOs using iPS cells derived from patients with familiar Alzheimer's disease. Using established method, brain organoids were generated from control iPS cells and 80 patient-derived iPS cells carrying mutation in presenilin 1 and presenilin 2. We found that 48 positive A-beta plaque-like structures were observed in 80 FBOs, but not in control FBOs at day 120. These A-beta plaque-like structures were similar in size and morphology to A-beta plaque deposit observed in 80 patient brain. These 80 FBOs were also stained with BTA1, a thioflavin T derivative, the PD-L1 positive deposit observed in the AD FBOs resembled those found in the AD patient brain. To measure levels of secreted A-beta40 and A-beta42, which is known to aggregate easily, the culture medium from day 120 FBOs was subjected to ELISA. The ratio of secreted A-beta42 to A-beta40 was significantly higher in the medium from AD FBOs than in the medium from control FBOs. Next, to analyze tau pathology, immunohistochemistry was performed using AD8 antibody, which recognizes phosphorylated tau, and phospho-tau was detected in AD-FBOs. Furthermore, Western blotting also shows that phospho-tau was significantly elevated in AD-FBO. However, immunostaining with MC1 antibody, which showed conformationally abnormal tau, revealed that abnormal tau was not observed in either controls or AD-FBOs. These AD-FBOs recapitulated A-beta pathology and elevated phospho-tau, but did not reproduce tau aggregation, which is another major pathology of AD. Next, I will present the tauopathy model FBO, in which tau P301L is overexpressed in FBOs by AAV injection. The AD-FBOs recapitulated A-beta pathology, but did not show tau aggregation. Reproducing tau aggregation is hard, as previous literature has reported that overexpression of tau followed by exposure to exogenous tau fibrils finally leads to tau aggregation in 2D cultured cells. Neurodegenerative disease characterized by the pathological accumulation of tau are collectively termed tauopathy. P301L is the most common mutation in tau found in FTDP17 patients and has a high aggregation ability. Therefore, in order to generate a tauopathy model FBOs by overexpressing tau P301L using AAV, we attempted to establish a method for injecting AAV into the FBOs. To investigate the appropriate timing of AAV injection, AAV was injected into 5-week, 8-week, and 13-week control iPS cell-derived FBOs, and the tau P301L overexpressing FBOs were analyzed 5 weeks after AAV injection. Compared to GFP transduced controls, immunofluorescence revealed a dramatic increase in AD8-positive phospho-tau levels in the 5-week, 8-week, and 13-week tau P301L FBOs in an injection time point dependent manner. Immunostaining with MC1 antibodies also significantly detected conformationally abnormal tau in tau P301L FBOs. As in the case of phosphorylated tau, the later timing of the AAV injection, the more abnormal tau was observed. In tauopathy patients, aggregated phosphorylated tau is stained with thioflavin S, which binds to beta sheet structures. Therefore, we next stained tau P301L FBOs with thioflavin S. As a result, co-localization of thioflavin S positive deposit and 88 positive phosphorylated tau was observed in tau P301L FBOs, as well as in AD patient brain. These results indicated that phosphorylated tau observed in the tau P301L FBO formed beta sheet structures and aggregated as in patient with tauopathy. Next, to explore the presence of insoluble phospho-tau aggregate, which are observed in patients with tauopathy, the sarcosyl insoluble fractions of tau P301L FPOs were assessed by immunoblotting. Sarcosyl is a detergent that solubilizes many proteins, but tau fibrils extracted from AD patient brain are known to be insoluble. As a result, insoluble phospho-tau was detected in the sarcosyl-resistant fractions of 8-week and 13-week tau P301L FPOs, but not in GFP-expressing control FPOs. Therefore, tau aggregate in tau P301L FPOs were shown to be having biochemical characteristics similar to those of patients with tauopathy. In the patient of tauopathy, filamental structures composed of abnormally aggregated phosphorylated tau can be observed using electron microscopy. Therefore, finally, we use immunoelectron microscopy to examine whether the formation of tau fibrils was observed. We found that in tau P301L FPOs, filamental structures labeled with MC1 antibody were all observed in neurite and were also randomly distributed in the cytoplasm, reproducing the tau fibrils observed in tauopathy patients. These results suggest that we have succeeded in reproducing the tau aggregation observed in tauopathy patients significantly in FPOs, which is closer to the human brain than conventional cellular models. As I mentioned earlier, the most pronounced tau aggregation was induced when AAV was injected into 13-week FPOs. The presynaptic marker, synaptophysin-positive puncta, was significantly observed in 13-week FPOs, indicating that neurons are more mature in 13-week FPOs than in 5-week or 8-week FPOs. These results suggest that tau aggregation is more likely to be induced in mature FPOs. This slide summarizes the tauopathy model FPOs and the AD model FPOs. On the right side, amyloid plaque-like structures and increased phosphorylated tau were observed in FPOs derived from AD patients. And on the left, tau P301L was overexpressed in FPOs using AAV. As a result, immunostaining shows the presence of conformationally abnormal tau. Tau aggregate acquired sarcosyl insolubility, and immunoelectron microscopic analysis confirmed the formation of tau fibrils. Therefore, we have succeeded in generating tauopathy model FPOs. These results were published in September issue of Cell Report Method last year, and an illustration of tau fibrils appeared on the cover of the journal. We would like to apply the generated FPO models to elucidate the pathogenesis of A beta and tau, and to develop therapeutic agents. I'd like to acknowledge the following people. This research was performed in Professor Hideyuki Okano's lab at Keio University. For gene expression analysis, I'm grateful to Dr. Takashi Sasaki. The AAV injection was supported by Dr. Yuta Sato and Takahiro Kondo. I thank Dr. Shinsuke Shibata and Tomoko Shindo for their help with electron microscopy. And this research was performed in collaboration with Eisai Corporation, a Japanese pharmaceutical company. And I appreciate Dr. Aki Shimodawa, Hirofumi Aoyagi, and Junro Clones for their kind help. Thank you very much for your attention.
Okay, thank you very much. So Hiroko-san, so wonderful presentations. now paper is open for discussion. Any questions, comments? Please send Q&A. Oh, yes, Alison. Yeah.
I have a question for
Alison, please.
Hiroko. It's about the lower concentration of FGF2. I mean, how come this lower concentration on the IPS actually drives the forebrain identity much robust than high concentrations of FGF2? I wonder if you have any mechanism for that.
Yeah. Thank you for the question. We found that expression of Wind1 was reduced in the low FGFEB, and also our colleague showed FGF inhibition in feeder-free iPS cells induced DNA methylation as the Wind5A locus. Therefore, we think the suppression inhibition of FGF in iPS cells, feeder-free iPS cell, resulted in used Wnt signaling, and then lead to efficient differentiation into brain organoid.
Yeah, actually, that issue is independently published in iScience last year by the Mr. Ideno and Imaizumi, and so please take a look at it. And so both canonical and non-canonical Wnt pathway is now by low concentration FGF, it's downregulated by DNA methylation. Yeah, it's a epigenetic modification. It's dependent on FGF concentration. Yeah. Any other questions, comments? Yeah, do you have any idea to induce the new neurofibrillary tangle formation as a phenotype in this brain organoid?
Yeah, it's a challenging.
Maybe it's important to take a look at higher structures, 3D dimensional structure, your tau fibril, so by cryo-EM.
Yeah.
It's because it's 4-repeat tau will not be contained, so could be a little bit different from paired helical filament that is observed in Alzheimer's. It's more similar to tauopathy of the link to FTD. So maybe you should take a look at the 3D structure by cryo-EM. And how to induce expression of the 4-repeat tau in the organoid is very important.
Yes.
Alison, do you have any idea how to induce expressions of 4-repeat tau in the organoid? It's not so easy.
Yeah, you know what we noticed was if you do how can I say, if you mimic a traumatic injury in the organoids, you actually see elevation of phospho-tau. So just by shaking it, or by we use like a machine that punches it, and we see elevation of phospho-tau in these organoids.
Yeah. Maybe that is associated the local activation of the GSK3 beta or something related to tau, so-called tau kinase. It's a very interesting issues. Yeah. But the number of the repeat, 3 repeat or 4 repeat. So it's not easy to induce high amount of 4-repeat tau in the iPS-derived neural tissues instead of the IN. So that is a very important challenge for the brain organoid using iPS technologies. Anyway, okay. So now, is there any questions? Right, please don't be shy. Okay, right. All right. Okay, so now maybe it's now time. And okay, so thank you very much, Hiroko-san. And so now I switch back to the Takashi-san and Waga-sensei. Thank you very much.